Have you ever seen a SCOBY? This gooey mass of microbes is what produces the deliciously fermented drink called kombucha. A SCOBY is a symbiotic community of bacteria and yeast growing in unison by the billions in what's essentially a very large biofilm. I grew this one myself. I'm pretty proud, but I won't lie to you, it is a little wonky. It smells a little funky, so that could have been because of the cultures I used. I'll show you my process, and together we can learn about microbial metabolism. So in particular, I want to talk about how microbes obtain energy to not only produce this drink, but also grow and divide and multiply into this magnificent SCOBY. However, there is so much diversity when it comes to energy mechanics in the world of microbiology that we're going to focus on chemoorganoheterotrophy. I know, it's a lot, but don't let that word scare you. Let's break it down. Organisms can be categorized based off of three main things: where they get their energy from, where they get their carbon from, and where they get their electrons from. When we talk energy, I want you to think ATP. So contrast a plant who gets its energy from the sun, specifically in the form of photons, to a human like me who gets my energy from breaking down chemical bonds from the food that I eat. What are the names for these two examples of different energy sources? I know you want to say autotrophs for plants and heterotrophs for humans, but resist it. Those terms do apply, but they're not referencing energy. It's actually referencing carbon source, and this is a common misconception, so let's just take a minute to explore it. All living things need a carbon source, which allows the organism to build precursor metabolites and eventually macromolecules. Plants are autotrophs because they get their carbon source from inorganic CO2, whereas humans are heterotrophs because their carbon source is organic. For example, think of the sugars that we eat. All right, so then what do we call an organism that gets its energy from the sun or photons versus an organism that gets their energy from breaking chemical bonds? Phototroph is the proper term for organisms that use light energy, like the cyanobacteria. And chemotroph is the term for those that use chemicals as a source of energy. Lastly, you can also consider electron sources. The reason capturing electrons is so important is that it is directly related to how cells harvest energy. Basically, without a source of electrons, cells can't power the machinery that keeps them alive. Organotrophs get their electrons from organic molecules. Think glucose, fatty acids, amino acids, etc. Lithotrophs get their electrons from inorganic molecules like hydrogen gas, hydrogen sulfide, iron, or ammonia. And there's really cool examples of this in microbiology. So bringing it back to our big term, chemoorganoheterotrophy means using organic molecules for energy by oxidizing them. The organic compound is also typically the electron donor. Cells can do this with oxygen if it's around, or without oxygen if it's not, as long as they have the right enzymes to handle the toxic byproducts of oxygen. Now, I think that's a pretty cool note, so I don't want to just breeze past it. Although oxygen is so vital to so many life forms, oxygen has a tendency to react into toxic forms, like hydrogen peroxide, for an example. So, if an organism utilizes oxygen, it has to be equipped to deal with it. Before we dive into it, let me show you what goes into our kombucha. First, I dissolve sugar in water before steeping some black tea. The sugar is plain cane sugar, or sucrose, which is a disaccharide of glucose and fructose. This is essentially just microbe food. Now, the tea aspect isn't just for flavor. It actually helps select for kombucha-adapted microbes. Black tea and green tea leaves contain compounds like polyphenols and even small amounts of amino acids and other minerals. Some microbes in the SCOBY can use polyphenols as substrates, but these compounds also act as mild antimicrobials, discouraging the growth of many unwanted bacteria while being tolerated and, again, even sometimes metabolized by kombucha's yeast and bacteria. And finally, we need to add our microbes to the mix. I used some store-bought kombucha, which should have active cultures of bacteria and yeast. It's really best to use something that isn't flavored, but I couldn't find one, so I just picked my two favorites, added a little of each, and hoped for the best. The yeast and bacteria in our little kombucha experiment here both utilize and metabolize sugars, but they do so in different ways. So let's start with breaking down the difference between cellular respiration and fermentation, and then we can go ahead and apply those concepts to our kombucha. Cellular respiration is how cells harvest lots of energy from substrates like glucose and store that energy in the form of ATP. Cellular respiration is an umbrella term for energy generation pathways that include glycolysis, if the substrate is glucose, the citric acid cycle, electron transport chain, and oxidative phosphorylation. If the process is aerobic, it means that oxygen is the final electron acceptor, and this gives the biggest ATP payout. But it can also be anaerobic, which means that something else, like nitrate or sulfate, is the final electron acceptor and oxygen is not involved. The process, though, is overall pretty much the same. While descriptions online will vary, fermentation is not technically considered cellular respiration because it doesn't follow that general pathway. In particular, it doesn't utilize the Krebs cycle or citric acid cycle, and there's no electron transport chain. Aerobic cellular respiration can start with differing fueling sources, but we often learn about it as starting with glucose. This is actually pretty helpful because once you learn the process, you can see how other starting fuel sources like lipids or even proteins can be broken down into smaller molecules that just wedge themselves right into that same pathway. So let's do a recap of cellular respiration starting with glucose. It all starts with glycolysis, where glucose is our starting molecule and the products are two pyruvate molecules, ATP and NADH. Note that in eukaryotes, this step happens in the cytoplasm, not the mitochondria. When going through the steps of cellular respiration, it really helps to think about that original glucose molecule and how it's being broken down along the way. Glucose is a 6-carbon molecule, and during glycolysis, glucose essentially gets chopped in half into 2 3-carbon pyruvates. Bonds between atoms are like a form of potential energy, and when you break those bonds, energy is released. You can also think of electrons as sort of like a unit of energy. Remember how earlier we talked about the importance of both energy, electrons, and carbon in these processes. So when the glucose is broken in half, electrons are also harvested in the process. But where do they go? NAD+ is an empty electron taxi. By the end of glycolysis, it gets reduced to NADH, meaning that it picked up some electrons. I like to visualize it as NAD+ picking up one electron, which I can visualize as pairing with that positive charge on the molecule to make NAD. And then it basically uses a hydrogen ion, or a proton, to grab onto one other electron, making it NADH with no charge. Pay attention to these electron taxis. You'll notice electrons being harvested throughout the process of cellular respiration, and you'll see that they all have the same destination regardless of pickup point. They all go to the electron transport chain. But before we get too ahead of ourselves, what happens after glycolysis? After glycolysis, pyruvate moves into the mitochondria. That is, if we're talking about eukaryotic microbes like yeast. If we're talking about bacteria, we're still in the cytoplasm. Each pyruvate is modified into acetyl-CoA, which kicks off the citric acid cycle, also called the Krebs cycle. Notice that acetyl-CoA is a 2-carbon molecule. One of these carbons was taken off of pyruvate and is now in the carbon dioxide produced during that step. The citric acid cycle breaks down our carbon-containing molecules even further, releasing carbon dioxide. And in the process, we fill up more electron taxis, NADH and FADH2, plus a little extra bonus ATP is made in the process. By the end of the citric acid cycle, we are done breaking down what was once a glucose molecule. Now, we utilize those harvested electrons to fuel our ATP factory. NADH and FADH2 taxis all head to the electron transport chain, which is embedded along the inner membrane of the mitochondria in eukaryotes. In bacteria, the electron transport chain is found on the plasma membrane. Here, high-energy electrons are passed along protein complexes. You can think they're unloaded from their taxis now that they're at their final destination. And the energy released as these electrons move through the chain of proteins actually powers those very same proteins to pump hydrogen ions, aka protons, across the membrane. The result is a hydrogen ion gradient, also called a proton motive force. Hydrogen ions, or protons, are pumped against a gradient so that there are more protons on the outside of the inner membrane than there are on the inside. By the way, where do these protons come from? Well, when our electron taxis like NADH release electrons, they also release hydrogen ions. Think of this gradient like a downhill slope, and protons are like water. Water wants to flow downhill, right? Well, similarly, protons flow down that proton or hydrogen ion gradient through ATP synthase, which spins like a turbine to generate lots of ATP. It's seriously like a hydroelectric power plant inside of the cell, and this is called oxidative phosphorylation. But there's something we need to resolve. So electrons get passed through a series of proteins in the electron transport chain, But when those electrons get to the end of the chain, where do they go? They need to go somewhere, or otherwise they would cause a traffic jam. So the final electron acceptor in this system is oxygen, if we're talking aerobic cellular respiration. Oxygen combines with electrons and protons, the very same protons that just flew down ATP synthase, to form water. If oxygen is around, many cells carry out aerobic respiration. But what if oxygen isn't available? Some microbes will use other electron acceptors. For an example, nitrate, and this would be called anaerobic cellular respiration because it's in the absence of oxygen. Some microbes can do both, and some can only do one or the other. In fact, some microbes that strictly use anaerobic respiration can even die on contact with oxygen. Interestingly, many of the yeast in kombucha, like for example Saccharomyces cerevisiae, can do aerobic respiration. However, in kombucha, they will preferentially use fermentation instead. These yeasts are displaying something called the Crabtree effect. When sucrose and glucose is abundant, they will preferentially ferment these sugars into ethanol, even in the presence of oxygen. This is essentially a make energy fast, not efficiently strategy. Fermentation is going to make ATP faster, but the yield is much lower than with cellular respiration. What's going on in fermentation? During fermentation, we start with glycolysis, just like we saw in cellular respiration. The microbes actually are basically just doing glycolysis over and over and over again because a little ATP is made in the process. In fact, glycolysis alone produces 2 ATP per molecule glucose, but compare that to the 28 to 32 ATP from full cellular respiration. But cells would run into a problem if they just did glycolysis over and over again with no extra steps. Remember that during glycolysis, our electron taxis, NAD+, get filled up or reduced to NADH. Well, those taxis would just keep getting filled up until we eventually run out of them. We need a way to empty the electron taxis so that the party can keep going. So, pyruvate from glycolysis can be converted into products like ethanol and CO2. That's actually why kombucha is fizzy and slightly alcoholic. Or some acids like lactic acid, which adds sourness. And in the process, it regenerates an NAD+, so that glycolysis can keep running. Another advantage of fermentation, besides speed and lack of need for oxygen altogether, is that the byproducts, acids, alcohols, and CO2, act as a natural preservative for the tea and might also feed some other microbes in the symbiotic community of the SCOBY. In fact, inside kombucha, we can find a beautiful example of something called metabolic cross-feeding. Acetic acid bacteria in the kombucha use aerobic respiration to oxidize ethanol into acetic acid, and gluconic acid and glucuronic acid. The acids they produce drop the pH of the tea solution. This helps the yeast because acetic acid bacteria prevent the buildup of too much ethanol, which can be really toxic to yeast. But how does this fit into what we learned about aerobic respiration? What I just described is only a partial oxidation. Electrons harvested from the breakdown of ethanol to acetic acid go straight to the electron transport chain, where we learned a proton motive force is generated and ATP is created via oxidative phosphorylation. A complete oxidation could take things further. Acetic acid could react to form acetyl-CoA, which you should recognize from earlier when we learned about aerobic respiration. Acetyl-CoA enters the citric acid cycle, and we see the full respiration of that carbon-containing molecule to carbon dioxide. This would generate a higher energy yield, but it wouldn't create the right acidity that the SCOBY has evolved to thrive in. For me, understanding the SCOBY as this mini ecological community of microbes working in syntropy, it makes sense that the acetic acid bacteria utilize an energy harnessing pathway that pumps out the acetic acid over maximizing ATP yield from complete oxidation. To be honest, I feel like we barely got to discuss the metabolic processes that can happen in kombucha. In a single jar, it feels to me like an entire planet of microbial inhabitants, and they're all just thriving and eating up sugars and co-creating this drink that so many people love. And actually, you can very strongly argue that humans are in a mutualistic relationship with SCOBY as a community, because as the SCOBY is fermenting the sugars in the drink, I am monitoring that, and I'm going to stop this fermentation process before too many toxic byproducts start to build up, like ethanol, for an example. At that point, I would take the SCOBY and transfer it to a new jar where it has an entirely replenished cache of sugars. So this SCOBY is getting a safe environment and tons of food, and in return, the humans get a tasty drink that they get to enjoy. So I hope you enjoyed learning about the nutritional types of microbes and some energy mechanics and metabolism, including aerobic cellular respiration, anaerobic cellular respiration, and fermentation, all through the lens of kombucha.